is exactly what we mean. If we compare the summary of species that were isolated from the environment and those that were isolated from the pigs, we take an example of Mycobacterium senusei. We see that in environment we isolated only one Mycobacterium senusei, and this is understandable given the dilution factor that we just talked about, that is controlled by the precipitation. We've isolated 24 from the animals, so that means the animals have more than what is in the environment. The M. terry also we isolated 1, but isolated 9 in pigs. The same thing. So there's a direct relationship between, in terms of species that are prevalent in the environment and those that are in the pigs, but they are much more amplified in the pigs. And this we think is because the pigs have to go around fending for themselves, looking for water and food, and in the end they end up accumulating this whole host of and of course the pigs that are housed indoors, for example in Europe, in Holland, they have a whole host of, they have different types of risk factors. In those areas they actually isolate a limited variety of mycobacteria, but in higher numbers, given that they are housed intensively, so they are definitely exposed to the same type. These ones, on the other hand, have a wide variety of mycobacteria isolated with them, so definitely it is a component of the management system that is responsible for the wide variety, but also the fact that pigs are concentrating it as they look for food. The third paper, we documented for the first time the incidence or prevalence of Mycobacterium bovis, the one that it causes bovine tuberculosis, that is in cattle. We found it in pigs, and pigs are supposedly spillover hosts. 2% of the pigs that we sampled had Mycobacterium bovis. We isolated this Mycobacterium bovis from a boar and 2 sows, and the lesions were characteristic of TB, of course, caseation and a grit texture with a deep yellow color. All these samples that were infected were from Madudu sub-county. That was we could document that. Now when we did the molecular analysis, we found that they were all affected by the same polygotype, SB1469. Now this polygotype had already been documented in the northeastern part of the Uganda cattle corridor, that's in Karamoja, in 2006 by James Oloya. Now we don't expect that the pigs in Mubende are actually making the journey from Mubende to Karamoja, so there must be something that is bringing this same isolate or this same genotype or to Mubende, or maybe we could say that this genotype is prevalent in the entire cattle corridor. It is highly unlikely. There must be some agent that is some host that is carrying them around. The most likely agent could be the wild pigs. For example, in Mubende around Lake Wamala, there is quite a number of wild pigs there, and those are known to move vast distances looking for food. They could be territorial in sometimes, they have certain territories, but depending on the season they do move around. And the cattle movement in the cattle corridor is quite high. It's a highly mobile area of human and animal, given the pastoral characteristics they have in this area. So we believe that the pigs are only getting it from another host that is normally supposed to carry it. Now, given that there's no study that is done on Mycobacteria M. bovis in cattle in Mubende district, we would use pigs as a sentinel, like an indicator of what probably is going on in cattle, and this would seem to indicate that actually the cattle are much more affected with the M. bovis. But given that there's no study, this would only be speculative. The fourth paper documented the characteristics of the isolated Mycobacterium avium specifically that was recovered in human, in cattle, and in pigs. Now we combine isolates from 2 studies. The study in the northeastern part from Karamoja, we used 12 isolates from human, 3 isolates from cattle in Karamoja still, and then the pig isolates that we isolated in Mubende, 12 loci from two studies that have been documented. The first study was in France that documented. We used 8 loci from France. These were the most allergically diverse loci that we picked, and then we also used 4 loci from Japan. So we put this together and made a tool for characterization that was based on 12 loci. Now this tool divided these samples in 2 groups that directly put them back into their geographical areas that were sampled. Geographical area or time difference, because the cattle and human in Karamoja actually shared genotypes. This would simply mean either the human are getting it from cattle, or cattle and human are getting it from the same environmental source, and it's more likely that they are getting it from the same environmental source, given that these pastoralists are, they share water sources with their animals. So the results look like that, and we have the pig samples and the cattle samples. We see that the cattle samples and the human samples share a genotype, and this would have public health implications. The tool, we use the best allelic diverse loci from Japan and France, so we'd expected these to be the best, and they would provide us quite a high discriminative power. But this tool gave us only an 88% discriminative power. Interpretation of this study simply means that you can't simply pick a tool that is developed from somewhere else and use it as a diagnostic tool or characterization tool on isolates from another area. So there definitely is a need. There is a need to develop geographically tailor-made diagnostic tools, especially in this area, if we are going to be able to trace where these mycobacteria are coming from, whether it's from the environment or the animals, especially for public health purposes. The human case study, the 5th paper, we documented molecular genotypes and drug susceptibility as well as economic aspects of tuberculosis in Mubende district. We found that 21% of the people we sampled actually had tuberculosis. Pulmonary tuberculosis was the most prevalent. 62% of the TB patients that we found actually were also co-infected with HIV, but there seemed to be a stratification in terms of age. Most of those who had a co-infection were within the ages of 20 and 30. Now, to take you back a bit about the population structure of Uganda, remember we said Uganda has one of the youngest population structures, with almost 50% of them living being below the age of 15, and that age structure is walking right into this age bracket that is highly co-infected. I'm not saying, if this is supposed to be extrapolated to other places, I don't think, in Uganda, I don't think that the picture would be much more different in terms of age structure of co-infection of TB and HIV. For example, for us, in Kampala or Masaka or any part of Uganda, I don't think that the age structure of the co-infected persons would be any different from what we found in Mubende district. And that would then simply mean that this young population is walking into an age structure that is potentially highly infected with the co-infection. We also found an association with smoking, and within the results that we got from the anthropological study, we found that smokers actually share cigarettes, and we believe that this cigarette sharing could be the answer to the association between TB and smoking. That an individual that is infected and sharing his cigarette, chances are is going to pass on this bacteria to the other individuals while they share. And we found through a random effects model that there was more variation in TB prevalence at sub-county level. That means every sub-county seemed to offer different dynamics to the TB epidemiology. And we see from the model that an HIV patient was almost 3 times more likely to have TB, or the other way around, a TB patient was 3 times more likely to be HIV positive. A TB patient was almost 5 times more likely to have come from a household that has between 6 and 10 individuals, and almost 4 times more likely to be a smoker. But on the economic side, we see that individuals spend more money on transportation. And this is evident of, you know, the long distances that these people have to move to access this medication, and the lack of equitable distribution of quality hospitals. Is one hospital that these people have to go to regardless of how far they live. Now imagine you have TB infected lungs. I mean, TB compromises how much? But the same lungs are the ones you're using to ride a bike to go and access medicine. So of course with time this is going to be a hassle to the patients, and the TB strategy, control strategy, is that find as many people who are infected with TB, who are positive to the PPD, who are positive to the X-ray, positive to the acid fast test, and all hosts of tests, and treat them as fast as possible in order to prevent them passing on the TB to other people. Now, if people can't access these hospitals where all these diagnostics are, that means you're going to catch fewer and fewer people. Therefore, this is bound to be the bottleneck to this current TB control strategy. The high medical related transport bills is bound to be the bottleneck. On a molecular picture, 52 isolates clustered in 12 groups, and majority of these actually belong to the T2 Uganda group. And T2, these are the modern types of TB. T2, T1 that is prevalent in Russia, T2 is prevalent in Uganda. What do we mean by this? And if we look at the dendrogram, we see that Uganda 1 and Uganda 2, and this belong to T2 lineage, were the most prevalent. These are the ones that were circulating the most among this population. But we also had the Central Asian strains, the Central Asia Kilimanjaro, that is most prevalent in Tanzania, and the Central Asian 1 Delhi, that is, of course, the name comes from, you know, the people from whom it is most likely to be associated with. But of course, with the high number of Asian communities in Uganda, of course, that also shows, you know, the molecular epidemiology is also representative of the migration patterns globally. But then we also isolated the most peculiar isolate, the Ural. This Ural gets its name from Urals region. This Ural region is found right there, and up until 2011 this isolate had been only recovered in Tbilisi and Moscow in Georgia. Then how did it make its way to Uganda? Now, whatever I'm going to show you now is highly speculative information. It has nothing to do with what the reality is. It's simply speculation, and it could be far away from why this isolate is in Uganda. There is a gold mine right there. I can't be sure. I don't know who owns that gold mine, but I know that the people who refine gold in Uganda are actually Russian. They're the only ones that have this license to purify gold in Uganda. The person from whom this isolate was recovered from is a 16-year-old student who lives in Mubende town council but hails from Magogo. That's the village where he comes from. This specific strain also was resistant to streptomycin. Now, does this information give any picture of how this Ural got to Uganda? I don't know. It's speculative information, but it simply shows how complex the tuberculosis epidemiology is in Mubende. That you have strains that are coming from thousands and thousands of miles